Hi everybody, welcome to this talk. This talk is about chemistry and Covid-19 testing. My name is Philip Drake. I'm a research scientist based at the University of Bradford in the School of Chemistry and Biosciences, and I'm also a lecturer in chemistry. So I'm going to start this talk by introducing myself, the kind of research I do. Then I'm going to talk about bioassays and how we can look for viruses, introduce viruses to what they are and how they work, and then I'm going to look at the DNA testing and immunotesting that we can do to look for viruses. So let's make a start. So this is me, Philip Drake. I'm also in the bottom corner here. Hello. I'd look at material chemistry. So I use polymer systems, material chemistry systems to try to find bacteria and try to find viruses. I develop something called point of care technologies. These are very simple chemical tests we can do in hospitals, at home, in the doctors, anywhere you are, to try to look for markers in the blood, to find things that are biologically relevant, things like bacteria, viruses, but also cancer markers, heart attack markers. I also work in magnetic fields and how we can use nanoparticles to try to cure cancer, but that's a different talk. I'm not going to talk about that here today. My contact details are p.drake@bradford.ac.uk. Feel free to contact me anytime. Google me. You can find out about the kind of work I do. There's lots of articles out there. My Twitter handle is Philip Drake @materialchemist. Again, so if you want to follow me on Twitter, feel free. And I'm happy to give talks about chemistry careers, research talks, science talks to all ages. So if you want any kind of talks like that, then contact me and let me know. I'm always happy to talk to people. So, bioassays. I'm interested in material chemistry. You might wonder, what has polymers? When people think of polymers, you might think of plastics, polystyrene, plastic bags. But really, life itself is based on polymer chemistry, or at least I think so. I'm slightly biased. But life is based on DNA, it's based on proteins. It's based on how these things interact. I look at surfaces. Polymer surface. Polymers are very long molecules and they have surfaces. When a chemical interacts on that surface and binds to it, it can change the surface slightly. And we can look at that change, develop sensors. So if a molecule binds on a surface, we shine a laser light onto it. The laser light interacts with the surface. Photons are scattered off. As a little change occurs, you can get a little readout that says, yes, this thing bound, and this is how much it was. And that's essentially all a bioassay is. It's looking for things binding and transferring that into a signal. So there's lots of ways we can do them. They're called bioassays because we're usually looking for something within a biological system. But it could also be that we're using a biological molecule, to do some testing. So it's not necessarily has to be a bioassay. It could be a chemical test, really, is a more general term. Some very common chemical tests you might have heard about are things like pregnancy tests. In pregnancy tests, you have a strip of paper inside a plastic tube. The liquid moves along the paper. It interacts with things and carries some molecules along, and they form little lines. And if the line appears, then you're pregnant. Or if it doesn't appear, you're not pregnant, or something like that. But it's a very simple test. It's called a lateral flow immunoassay. Things are flowing along surfaces. We can use a very similar test to look for viruses too, and I'll go into that later. Another kind of chemical test is the breathalyzer, things police use. You breathe down a tube. In this case, the test is looking for ethanol. It's slightly different. It's not a surface interaction, but now when you breathe down a tube, the ethanol triggers a chemical reaction, and this chemical reaction creates an electrical signal, which gives a little readout on a screen that says how much alcohol is on your breath. Other examples of chemical tests are used by the police looking for drugs. You could be looking for heroin, cocaine, cannabis. These are very simple chemical tests looking for these molecules, and you'd be looking in different things. It could be looking in hair, in saliva, in urine. I do a lot of lectures on controlled substances, how we can detect them in different materials. So hair, I think, is quite an amazing thing. You can shine a laser on a piece of hair and you can dissolve that up and you can get little signals to say how long you've been taking this drug for or when it was in your body. They also use things like DNA fingerprinting. So DNA, all life uses DNA or RNA. Viruses actually use something called RNA, which we'll go into later. But DNA fingerprinting, everyone has a different sequence of DNA. the police use DNA profiling to see who's been at a crime scene. You take a little sample of blood, has some DNA in it, you can then link that back to a particular person. But it's not just people. Bacteria and viruses all have their own unique codes of DNA as well. So we can use very similar technologies for looking at identifying viruses. And then in the hospital setting, we have lots of medical tests, which are again based on bioassays. So blood tests, looking for biomarkers, looking for molecules that are linked to cancer or heart attacks or strokes. Another classic example would be diabetes, looking for blood glucose. It's a little chemical test that has an electrical signal in the blood. If the blood has got lots of glucose in, you get a very strong electrical signal, which gives you a little peak off, which tells you how much glucose you have, and you can monitor your glucose. So that's a very good example of a bioassay. So I work across all these different fields, looking for how we can look at viruses, how we can detect cancer, how we can use DNA for looking at viruses. So that's the kind of work I do. So let's now look at what a virus is. We're going to develop a test for a virus, so you need to understand a little bit about how viruses work to be able to do this. So here, welcome to influenza. This is the influenza virus. It's very similar to Covid-19 virus. It's a round spherical virus. It's got proteins on the surface. In the middle here, it has its RNA. So RNA and DNA are very similar things. RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. It's very interesting when you look at evolution. We have a theory that life actually began in the RNA world with RNA and then developed later into DNA. DNA is a lot more stable, and so it changes a lot less over time. So it's a lot more useful for genetics, carrying genetic information from generation to generation. However, RNA can mutate very quickly. So it's good for systems like viruses that want to adapt to different environments. I'm getting sidetracked. RNA, DNA, polymer systems, they're all amazing. So viruses are a very good example of what we can do with nanotechnology. This is an area I work with, nanotechnology, looking at nanoparticles. Viruses are very simple. They have their RNA in the middle, they have a coating surrounding that, and then on top of that they have proteins. These proteins act like little keys to open doors, and they can tell where the virus is, what kind of environment it's in, and then trigger a response. They're not all round, they come in different shapes and sizes. We have the polyhedral adenovirus, we have a long tubular helical tobacco mosaic virus. Again, this is the influenza virus. It's a different way of drawing it. Same as this one here, it's a round thing with blobs on the surface. This is one of my favorite viruses. This is the phage virus. This actually interacts with bacteria. So humans suffer from viruses. Animals also suffer from viruses. Trees and plants have viruses. And even little bacteria have their own set of viruses which work with them. So the bacteriophage works exactly as you think it would work. These little legs have little proteins on the surface and they can recognize certain bacteria. The phage will land on the bacteria on the surface and then inject its RNA through the bacteria, through the cell wall, and change the bacteria from an E. coli, for example, to a virus factory. So all viruses need to do is deliver their RNA into a cell. And they need to recognize what cell it is and open the door to that cell. And we can use these. We can take out the RNA, for example, and we could put in an anti-cancer drug. Then you have a virus now delivers anti-cancer drugs to cancer cells. So there is a lot of research in looking at how we can actually use viruses to deliver medical technologies. So viruses are amazing. This round thing here is a cell. It's a representation of a cell. It's a big ball of jelly. And on the surface, it has proteins. And the cell uses these proteins to talk to things on the outside. They have little doorways they can open up and interact. A virus is looking for a particular cell. It's a cell which is adapted for, which it knows it can infect. And it recognizes cells by these surface proteins. So these surface proteins interact with these proteins. They recognize each other. They're like little keys. So these are the keys. These are the doors. There could be red doors. So the virus has a key to opening all red doors. It floats off down a street, tries all the doorways. When it finds one that opens up, it can go into the cell. It opens the door up and comes in. When it's inside the cell, all it needs to do now is just open up and release its RNA. RNA are like the programs which the cell uses to make proteins, like little books and codes. These go into the nucleus. The nucleus is the central machine, the brain of the cell, which makes things. Oops, moved on too far. So the nucleus takes the RNA from the virus. It reads it and does what it's told to do, which is to make more virus proteins. So the cell starts to make virus proteins. It makes more copies of the RNA as well. The cell surface changes from having the cell proteins to having virus proteins on the surface. The RNA that's made is all packaged up into a tight ball, like this ball is here. It then bubbles out to the surface. As it leaves, it picks up its new proteins which it's made and becomes a whole new virus particle. This virus particle then goes on to infect another cell. So that's how viruses work. They go from cell to cell. So this cell stops being a heart cell or a lung cell and starts to be a virus factory. And that's where the damage comes. If too many viruses infect the lung cells, then all your lung cells start to die away, and you can't breathe properly. So that's where the damage comes from. So how big are viruses? I work in nanotechnology. Size is important. I work with small things. Viruses are about 100 nanometers. How big is 100 nanometers? This is the human hair. Don't know whose it is, some skin with a human hair sticking out. This hair here is around 100 micrometers in size. If we look at that under an electron microscope, we can see the human hair, we can see the thickness of the hair. We know that that's about 100 micrometers. This little scale bar here is 10 micrometers. We can see details in the hair. It's an amazing thing, electron micrographs. Again, that's another talk. This is a red blood cell. We know red blood cells are around 8 micrometers, so about the same size as this scale bar here. So this is a red blood cell drawn to the same size as the hair. Zoom in on the red blood cell, 7,500 nanometers in size. It's around 7.5 micrometers or 7.5 times 10 to the minus 6 meters. So this is the red blood cell. This little dot here is a virus particle. It's about the same size as my pointer. That's how big they are. They're tiny little things. This little particle goes into the cell and infects the cell. So this is COVID-19 virus. This is what it looks like. It has proteins on the surface. It's a round sphere. We get this picture. This is a perfect model of what we know so far about the virus. We get this from looking at electron micrographs. These are some pictures of some real COVID viruses. We have a little corona around the surface. This little corona is made from these proteins. So that's what they look like. We take thousands of pictures of the virus and put them into a computer. The computer looks at all the images and models what the virus looks like. And this is where we get this detail from. We can even see inside here. So this is inside a virus. This is the RNA inside. On the inside is what it looks like. All this RNA is coiled round into a big circle. And that's what we see here. The shell and the RNA. So that's what viruses are, what they look like. This is the same virus. It's different color code. So here we have a purpley color code for stuff. Here we've got reds and whites, but it's the same picture, the same proteins. There are spikes on the surface of the virus. Look of a corona surrounding the viron. This is viewed by a TM. In this view, the protein particles E, S, and M are located on the outer surface. So a novel coronavirus named Severe Acute Respiratory Syndrome Coronavirus 2, SARS-CoV-2, was identified in 2019. And the illness caused by this virus is called Covid-19. So that's the virus. That's what it looks like. That's what we know. These are some real pictures of the viruses. This is the first patient in the USA that was infected with the virus. When you have a new virus, you have to find it. So this is what people are looking for. This here is a real virus coming out of the cell. This is a cell, this is the virus. If we look at our model, that's the model of the virus. If we turn that to black and white, you can see how this is these. These are the virus particles, and this is the cell. So that's what a virus is. That's how it infects a cell. It comes out. Once it gets to this point, we can start to sequence the RNA. We can develop tests for it. So the next bit part of the talk is going to be looking at these tests and how we can actually test for the virus. Okay. So let's look at the timeline of infection for the COVID-19 virus. Why is that not moving on? Here we go. So this is the start of symptoms, day 0. 0 is when you start to feel ill and you start to show symptoms. You're actually infected with the virus a few days before that, so the virus enters your body and starts to replicate. So the number of viruses in your body starts off at 0 and increases. It grows and grows and grows and grows. It reaches a maximum just after you start to feel ill. This is when you start to feel ill and you get lots of viruses in your body. At the same time, your body responds. It starts to detect the virus and has an immune response to that. So it starts to detect that down here. Before you have symptoms, your body knows you're ill and starts to fight it. It fights it with antibodies, so it starts to produce antibodies here. The antibody production is slow at first and increases and gets very rapid and makes lots and lots of antibodies. So as the antibodies are made, the virus then starts to be destroyed. So the virus concentration reaches a peak and then drops down. It goes down and down and down until eventually it's disappeared. You no longer have the virus. But the interesting thing is antibodies. You start to make them. When the virus is gone, you keep the antibodies in your blood. They float around waiting to be attacked again. If the virus comes back in again, starts off here infecting you again, you don't have this long build-up for antibodies. They're already there. The body fights them off quickly. The virus can't take hold and disappears, doesn't make you ill. So this is how the timeline of infection looks. If there's lots of virus in your body, you can detect them using nucleic acid. You can detect them using DNA. You can find the virus, you can extract the DNA, you can identify the virus. So in this window here, from symptoms beginning to about a week after, you can detect the virus using a PCR test, looking for its DNA. Another way to detect the virus is to look for the antibodies, but you can't do that straight away because you don't have any antibodies in your body, in your system. So after about a week after symptoms begin, you can do an antibody test and see if you have the antibodies in your blood. And if you do, then you know you've been infected. So the antibody test works about a week after your symptoms begin. But the PCR test could actually work before you even know you're ill. So a few days before you know you're ill, you can still do a PCR test and find the virus. So let's look in detail now at the DNA test, how it works. DNA testing has lots of different names. DNA profiling will do, but in terms of science, we have nucleic acid amplification test, the NAT test, and a polymerase chain reaction, the PCR test. DNA is made up of nucleic acid. It's a polymer chain of nucleic acids all put together. So you need to know a little bit about the structure of the DNA. Kerry Mullis won the 1993 Nobel Prize in Chemistry. So chemistry researchers that were developing these systems. So DNA is a double-stranded molecule. There's two DNA coils, alpha helix, going around. So we can illustrate those with a red strand and a blue strand. These are different, they're not the same, they're complementary. So they bind to each other in a complementary binding system. So we have a red strand and a blue strand. If you know the DNA sequence you're looking for, so if you know it's a COVID-19 virus, you can sequence the DNA, you can make synthetic bits of DNA. We call these little primers, a forward primer and reverse primer. So we have a little bit of blue DNA which will bind to the red sequence, and a little bit of red DNA which will bind to the blue sequence. So if we take the DNA, say we've got one virus particle in the blood, we extract the DNA from that, we have one strand of DNA, actually RNA to start with, but we can convert RNA using an enzyme to DNA. So we have one strand of DNA. If we denature DNA, it means we pull the strands apart. So we simply by heating it up, we can make these two strands come apart and break off. So we denature the DNA, gives you our two strands. Here we have double-stranded DNA. Here we have two single strands of DNA, the red one and the blue one. Heating the DNA up goes to here. If we then add the primers, the synthetic sequences we made, we know this sequence, we know the codes of these points, so we can make little synthetic pieces. We add those into this mix. Once you start to cool it down, the DNA will then rebind to itself. If there's only one strand of these two and millions of these, the chances are these are going to bind before that does. So your little primers bind onto the targets. They only bind to the exact sequence they're matched to. So these primers are made to only bind to COVID-19 DNA sequence. We call this annealing, sticking, rebinding. So your primers bind. We then add in an enzyme. It's a special enzyme, it's PCR enzyme. This little blob of protein looks for double-stranded DNA here, and it then reads along and it replicates and rebuilds the missing part of the DNA, called extension. So we add the primer, we add the PCR mixed, we get extension occurring. We rebuild this missing bit of DNA, which then results in two strands of DNA, exact copies of each other. So we start off with one strand, we end up with two strands, exactly the same. This is how PCR amplification works, how DNA profiling works. You start off with one strand of DNA. This whole cycle of heating, cooling, heating takes about a minute. After 1 minute, if you start with one strand, you have two strands of DNA. After 2 cycles, 2 minutes, you've doubled it again, you get 4 strands. You keep doubling all the time. After 3 cycles, you have 8 strands of DNA. But only the exact sequence, only the COVID-19 DNA is being amplified because that's what your primers look for. After 4 cycles, you got 16 strands. It keeps doubling. After 10 cycles, you got 1,000 strands of DNA. Typically in PCR, we do about 20 cycles. So after 20 cycles, how many strands of DNA do you think you have? 2,000, 10,000, 100,000? You actually have over 1 million. 1,048,576 strands of DNA after just 20 minutes of reactions. Over 1 million strands. We can use a special dye. It's called cybergreen dye. Cybergreen. This dye only fluoresces when it's inside the double helix. So outside the double helix, it's just a dye, there's no color. When it gets inside the double helix, it fluoresces green and gives off a signal. So we use this dye to then look at how much DNA is present. Bing, it's glowing. So this is the chemical structure of cybergreen. It's a nice flat molecule. It slots itself within the double helix. So when it's inside double-stranded DNA, it fluoresces green. When it's outside, it doesn't fluoresce. The negative test, there's no virus RNA, and so there's no DNA. So from the cybergreen, there's no fluorescence. Nothing happens. So we plot a little graph, fluorescence against number of cycles. The fluorescence, if there's no virus present, the fluorescence just stays flat. Nothing happens. It stays at around 0. It's a negative test. If you have virus in your blood, you get a positive test. The RNA from the virus is converted to DNA. The concentration of the DNA increases every cycle. It can be detected. So then, as it's increasing, the fluorescence increases. So if we plot cycle number, at the beginning, you get very little bits, 1, 2, 4, 16, 32 strands. Eventually, you get lots of DNA forming, and the fluorescence goes up in this little path, the little shape, S shape, a certain cycle number. That's a sign of a positive test. We can also set a threshold value and a CT value. So we look at when the fluorescence goes above this point, then it's a positive test, and that tells you how many cycles you took to get to that test. So if there's lots of DNA at the beginning, your CT value is down here somewhere. If there's not so much, it's further down. So we can use that to actually quantify the amount of DNA. So this is the results from one of my papers. This is exactly the same plot we just did. We have the cycle on the bottom, we have fluorescence intensity, and I was looking for Staphylococcal aureus, MRSA, pathogens. And we can see the fluorescence follows this path and increases, and how much we can detect. If you have 1 times 10 to the minus 14 moles of DNA, you get your CT values very low, less than 5. If you got 1 times 10 to the minus 20, it's a bit higher. So we can quantify the DNA. It's a very simple test. It's just looking at the fluorescence increasing. So that's basically how the coronavirus test works, looking for PCR. You take a swab of the inside of the patient's nose or back or throat. Sample is then tested in the lab. Take the swab. If the swab contains the virus, you have the virus RNA, you can extract it. The RNA of the virus is extracted and purified. An enzyme, reverse transcriptase, converts the RNA to DNA. So you go from an RNA sequence to a double-stranded DNA sequence. The DNA is mixed with primers. These are synthetically made. We have to know the sequence. So you have to detect the virus first. So we have to find it, sequence it. Once we know the sequence, the RNA sequence of the virus, we can develop these tests. So we synthetically make a little bit of DNA which recognizes the virus DNA. Nothing else, only that will be amplified. So if it's present, amplification occurs, you get loads of DNA, fluorescence increases, you get a positive test. Again, positive test, you get this S-shaped curve, fluorescence increasing. A negative test, fluorescence just stays around 0. So that's how the DNA test works, the PCR test for a virus. I got this information from Compound of Interest, Compound Chemistry. They have a very good Twitter account. They have lots of good information about chemistry, so do go to their sites and have a look at those. Next, I'm going to talk about immunoassays, how they work. So that was DNA testing. Now let's talk about antibody testing. Covid-19 coronavirus antibody serological test. It's a blood test. It's designed to detect the antibodies. These are also called immunoglobulins, IgG and IgM. We're going to go into detail of these later. Detects the immunoglobulins against the coronavirus that cause the disease COVID-19. So antibodies are proteins produced by the immune system in response to an infection, and they're specific to that particular infection. First of all, we need to understand a little bit about what antibodies are. So this is an antibody. It's a Y-shaped molecule. We often represent it with just a Y shape. They're actually slightly different. So we have an FC component. This little chain down here is slightly different, and then these two are identical. And it's the end of these that detect certain structures. So the shape of the end of these can change, and they can stick to complementary shapes. So again, it's like a lock and a key theory. The virus you're trying to detect is the key, and here we have a lock and it fits exactly the same shape. So in the Y, we have these two lobes are the same, this one's slightly different. All human antibodies have the same sequence down here. They're all different up here because they all detect different things. So your virus has the proteins on the surface. The virus is called the antigen. Your antibodies have got these two ends, and these little white bits here are what recognize the proteins on the surface of the virus. So these ends will stick to the protein, and they're very specific. They look for a particular protein, nothing else, that has the right shape, the right key to these ends, and they stick to them. So that's how antibodies work. They label antigens in your body. If you have COVID-19, you'll have antibodies in your blood. Here we are. This is IgG, so the green antibody. This recognizes COVID-19 virus. These antibodies bind to the proteins on the surface of the virus. So this is the virus. These little proteins stick off. Again, these are what the virus uses to open the cell, but we can also recognize those with antibodies. So your antibodies come zooming in. They recognize these and they stick to them. So we can test for these antibodies using a lateral flow immunoassay, very similar to pregnancy tests. For this, we need a few special components. We need some chicken antibody that recognizes human IgG. Why do we need chicken? Find out in a minute. We need a label, something we can detect. It's often a gold nanoparticle, so nanotechnology can come into this. It could also be a fluorescent molecule or a dye molecule. It's just something that has a color that we can see. We have an antibody that recognizes chicken IgG. So again, why do we need that? Well, we'll find out in a minute. We also need some virus proteins. So we need to know the structure of these proteins. Once we know the structure, we can synthetically make them. And once we make them, we can start to develop tests for them. So we need some virus protein. If we have these four components, then we can make a test for coronavirus. Lateral flow immunoassay. It's a long strip of paper. We place some capture antibodies and virus proteins onto the surface. So they're stuck on here. This is your anti-chicken IgG, stuck on the surface. And this is your virus protein, the star shape, stuck on the surface. So your anti-chicken, every mammal produces its own unique type of antibodies. So chicken, goat, mouse, humans. This little bit here, they can always the same depending on what animal they come from. So this antibody will recognize any chicken IgG. We have some reagents, and we store those at the start of the strip. The reagent is labeled with a dye, often a nanoparticle. So this is our reagent. It's chicken anti-human IgG with a nanoparticle label. This is free to move. So it's chicken anti-human. It's made by a chicken, but it will recognize any human IgG. And we stick a gold nanoparticle onto it. So if we've got these, we've got our capture bodies, antibodies bound, and the protein bound on the surface. We have some reagent that's free to move on a strip of paper. We put a bit of blood onto this strip of paper. If the blood contains Covid-19 antibody, it's going to give you a positive test, you'd hope. So that's the Covid-19 antibody, the green antibody. Remember, these are human antibodies to Covid-19 virus. That's what it's labeled up here. So the blood flows down over these. As it flows down, it picks up this reagent, interacts with it, and keeps on flowing until it gets to this point here and it interacts with these. So the blood flows down the lateral flow system. If it's a positive test, you get two lines. You get some color appearing. This point here captures this dye, and this point here captures this dye. So you get two lines forming. The antibody has recognized the virus from the protein, from the protein from the virus. Your label recognizes human antibodies, so that sticks to that. This is recognizes chicken IgG. Your label is made from chicken IgG, so that sticks to here. So you get two lines. So in a negative test, you don't have any antibodies. So you've got the same setup. The blood just flows down, comes trooping down here, flows down. There's no virus antibodies, so nothing sticks to the virus protein, but your capture still captures the label, so you still see a line appearing. So this is called a negative test. Only one line will appear on the strip. So for a positive test, you get two lines. For a negative test, you get one line. It's really important to have this negative test. Whenever you do experiments, you've got to have a control experiment. Imagine that you've stored this reagent in the light, and this broke down, disappeared, no longer worked. So you'd think nothing would happen. You get no lines appearing. If you get no lines appearing, you'd think you might have a negative test, but you weren't. It wasn't that you were negative, it's that your reagent had broken. The test itself didn't work. So this control line is there to make sure the test still works and gives you a positive result. So positive test, you get two lines. It could be two equally thick lines, or it could be a really strong line and a very weak line, but it's two lines positive. One line is negative. Anything else, no lines at all, or just a positive on your test line, then that's invalid. It means the test didn't work. And it's really important to have that control. So you take a drop of blood, you mix it together, you drop it onto the test strip, you wait a bit, two lines appear, positive test for coronavirus. One line appears, negative test. So we can go a bit further. This is the same graph we saw before about the timeline of the infection. There's a lot more detail on this, so this gets a bit overwhelming, but the virus can be found in different parts. But what's important is this bit here, these two lines. This is something called IgM, and this is IgG. So we make two different types of antibodies. One is IgM, and one is IgG. And the IgM actually disappears over time. So we detect both of these different types of antibodies, we actually get more information. So what is IgM and IgG? Well, we saw IgG, it's a Y-shaped molecule. IgM is actually a pentamer of this. It's much bigger. It consists of 5 of these stuck together. This is slightly different. This sequence here is slightly different, stuck together, but the ends are the same. These are the ends that recognize the virus. This is exactly the same, but there's 5 together. This is your IgM, this is your IgG. So we add a third line to the test strip that now responds to IgM in the blood. This can tell if a person had a very recent infection, so it gives you a lot more information. So same again, we've got a test strip here, and now we've got 3 lines. One detects IgM, one detects IgG, and you still have your control line here. So you get 4 results. Negative test, you only get 1 line, control line. That's a negative result. You might have IgM positive. That'll tell you something. You might have IgG positive. That will tell you something else. Or you might have all 3. And that again will tell you something else. So we get 4 different results, a lot more information. This is what it actually looked like. There's some real test strips with real lines appearing. So, turns out we make IgM first. IgM starts to be made first. It's the first response from the body, is IgM. But interestingly, IgM disappears once you've cured the illness. Once the virus has gone, the IgM will disappear. IgG is what we keep for our immune response forever. So if we test positive for IgM but negative for IgG, you know that the infection has occurred within the last week. So the person's infected, they've got antibodies, but they've only been infected for 1 week. If you test positive for IgG only and not IgM, you know you had the infection, but it could have been a year ago. It could have been 6 months ago. It could have been 2 months ago. So you had the infection a while ago. If you test positive for both of them, IgG and IgM, you know you're in this region here. You've had the infection fairly recently, within the last month. So we know if you've had it within a few weeks, within a month, or you had it sometime a while ago. So adding the IgM gives you a little bit more information. And that is how antibody tests work. Any questions? If you do, ask them now. Okay, bye-bye.